So my name is Hannah Bryan, and today we'll be talking about protein techniques to address neurodegenerative diseases. We'll be talking about what are neurodegenerative diseases, the molecular basis of these neurodegenerative diseases, and how those molecular bases can be used and new techniques to prevent or at least slow down the rate of degradation within these disorders. These disorders can be very, very detrimental to the quality of life that the individuals suffering from them have, and so it is very important that these sort of techniques are studied. Here we got an example of the different hallmarks of neurodegenerative diseases from neuronal cell death, and then, which is obvious, we see the neuronal cell death within neurodegenerative diseases, and then the pathological protein aggregation, which we're going to be looking at some techniques that can help prevent the aggregation or go back and minimize the aggregation that has already occurred by getting rid of those unwanted proteins. We see inflammation, and inflammation is a key one because it can really be the catalyst to continue this degradation system, because as one cell dies and it's being killed by this disease, it is not following the apoptosis pathway that a cell normally should, and so those mechanisms and tools that should allow the apoptosis to occur aren't being recruited, and so the cell is just experiencing necrosis, and as necrosis occurs, it's making this cell explode and cause inflammation to the areas around it, causing damages to those as well. And this is very important to note because it is one of those things that can propagate the continuation of these diseases. So what is a neurodegenerative disease? A neurodegenerative disease is a type of disease in which a cell of the central nervous system stops proper functioning or dies. These diseases progress over time and currently have no cure. They have multiple different potential causes, from a genetic predisposition to the onset being caused by a tumor or a stroke, or from a history of abuse of alcohol or other substances. These chronic conditions damage or destroy parts of the nervous system and cause impairments to cognition, learning, and movement. Currently, the main goal is to stop or slow down the progression of these diseases with the treatments that we have. The main types of the diseases are dementia-type diseases, and these cause the progressive damage to various areas of the brain and cause neurons and several structures to die. These include Alzheimer's, frontotemporal dementia, chronic traumatic encephalopathy, and Lewy body dementia. Another one would be demyelinating disorders, and we can look at this example right here of a healthy axon with a healthy amount of myelin. What the myelin does is the myelin allows these signals to go down the axon quicker. It also acts kind of as a shield for the axon, because as we see all the way down here, whenever there's the disruption of axon function, the axon itself is being damaged because of the lack of myelin. The myelin allows these signals to move quickly, and so that's why we have that instantaneous reaction almost with getting the interaction with stimuli. Whenever you're no longer able to have that myelin there, the axon is no longer able to properly function. Examples of these would be MS in neuromyelitis optica spectrum disorder. And here is where we see an example of muscular sclerosis, where this axon is healthy with a healthy amount of myelin, and you can see that this neuron is able to properly function. While this neuron over here, you can see there's damages done to the myelin throughout the axon, and it is unable to properly function. And you'll see this with the impaired physical ability of a person suffering from MS. Another type of disorder would be Parkinson's disease, and it happens because the damage to specific neurons in your brain that help manage coordination and precise control of muscle movements. These include Parkinson's disease, obviously, but as well as Parkinson-like conditions that share a lot of symptoms and similarities with Parkinson's. Then we have motor neuron diseases, which happen whenever certain areas that control your motor movements die as these degrees progress, and this would be an example of this would be ALS. And finally, prion diseases. Prion diseases work as one protein aggregates, it touches the next protein and mutates that one as well, and these spread very, very rapidly through the brain and cause that damage to move very quickly and tend to be pretty quickly fatal for the person suffering. The people that have prion diseases tend to die within the year of being diagnosed with it. An example of prion disease is mad cow disease, and another example that's more common is the Creutzfeldt-Jakob disease, and this one has a little bit of a longer survivability. However, it is from 1 to 10 years. Now, why is this type of research important? It is because while this is not a very common disorder, neurodegenerative disorders are not incredibly common. However, it is likely that you know a person who is suffering from a neurodegenerative disorder, or have at least known of someone prior, especially with Alzheimer's being as common as it is within our elderly population. These are mostly affecting the elderly, but younger people are able to be affected by them, and over 50 million people worldwide are affected by a neurodegenerative disorder. The World Health Organization projects that the number of people 65 and above within the next 30 years will double, and thus the amount of people suffering from a neurodegenerative disorder will also double, which makes it even more imperative that we have these sorts of treatments going on in the progression of the treatment plans that we have available. The symptoms of neurodegenerative diseases vary widely amongst the people who suffer from them, but this is mostly because everybody has a different brain. So the connectivity between two different people is always going to be slightly different, and thus how the damages occur is going to be different as well. Within dementia type, these symptoms experienced are confusion, memory loss, trouble thinking, and behavioral changes. Here we can see the physical and molecular level of an Alzheimer's patient. We see this healthy set of neurons over here. And over here we can kind of see these plaques that are forming. We're going to talk about the plaques that form at a later part of this presentation, but those are very indicative of Alzheimer's. They can either be, these are either plaques or they're neurofibrillary tangles, which are also very indicative of these disorders. And then again, we'll see, this is another thing that we'll talk about is the tau propagation within Alzheimer's disease. But you can see that there's this aggregation of tau, and therefore this neuron does not look identical to this one anymore. And we see that the damages are making this neuron not very functional. With demyelinating disease, common symptoms include tingling or numbness, pain, muscle spasms, muscle weakness and paralysis, coordination issues, and fatigue. Within Parkinson's, these often include slowed movements, shaking and tremors, balance problems, shuffling steps, and a hunched position. Motor neuron diseases are more general because it just depends on what areas of the brain are being affected. As the areas of the brain are being affected, the people lose those abilities associated with them. And then, as I said, the prion diseases, it is just so rapid that you just have this massive cognitive decline until ultimately the prions make it so that the person is no longer able to live on their own. Like I said, the amount of variation within these symptoms is just simply because every brain is different. So even if I were to hit the same disorder or dysfunction within a certain brain region as someone else next to me, the damages being in the same area wouldn't necessarily mean that we would experience the exact same symptoms, as the connectivity that we both had was very unique to ourselves, as your brain is kind of like a fingerprint, so everything is just completely unique to you. Although they are similar and we would experience similar dysfunctions, we would ultimately have different things going on because of the fact that we have different neuroconnectivity. Now, what are the causes of neurodegenerative diseases? The causes vary so drastically. As we see, the genetics play a role, your lifestyle plays a role, and your habits play a role as well. Most of them cannot come down to a singular cause. There's different ways that you can see that somebody had a genetic predisposition, because Alzheimer's is an example of one that is very common to be passed amongst families, and there are heavy family ties within it. Age is a really big factor that contributes to it, as most people who suffer from neurodegenerative diseases are elderly. The environment can also play a role, though, and one example of that that's kind of interesting to see is that there is a link between vitamin C and an increased risk of developing it. So if you have a lack of vitamin C, there's actually an increased risk of you developing a neurodegenerative disease. And this can be seen between a correlation of people who live further away from the equator receiving less vitamin C and having an increased risk of a neurodegenerative disease later on in life. It can also be impacted by your medical history. If you've suffered from a massive damage to your body or cancer or a certain type of infection, you can be predisposed to have a higher likelihood of getting one of these disorders. And then your habits, like I said, lifestyle plays a role. Diagnosis and testing. One way that we can test is through blood samples, but we can also use imaging techniques such as MRI and CT scans. And we can use those blood tests to see genetic predispositions that people may have, and then we can use these to watch the progression of the disease or decide that somebody has just massive atrophy within it and that they are most likely suffering from an NDD. Here we see a healthy brain versus an Alzheimer brain, and you just see just the mass. If you look in these regions that the arrows are pointing to, you just see this mass amount of atrophy that this person is experiencing because of this disease. And then there are some diseases that we are not able to find prior to death, and so we use histopathology afterwards, and these would include Pick's disease or chronic traumatic encephalopathy, which aren't diagnosable while alive but can be seen in a sample under a microscope. Management and treatment of these all is that they are incurable, and the best course of action is to try to prevent them from causing more damage. Due to the lack of this knowledge in this area, there's a need to determine new approaches to fight these issues. What we'll be looking at is different types of novel protein techniques, and these techniques are aimed at preventing the neurodegenerative diseases and focusing on manipulating protein folding, aggregation, and degradation pathways. Some ways that they do this is by enhancing chaperone function, stimulating atrophy, targeting protein-protein interaction, and using small molecule inhibitors to disrupt misfolded protein aggregation, or protein replacement therapies to deliver functional proteins to replace those that have been affected by the diseases. Within my thesis, the techniques I will be focusing mostly on will be techniques addressing Parkinson's and Alzheimer's. So, what is Alzheimer's? Alzheimer's is a complex neurodegenerative disease with multiple pathways and molecular targets under review to investigate to attempt to find potential treatments of protein-protein pathways. Primary targets include the amyloid beta aggregation, tau hyperphosphorylation, neuroinflammation, and synaptic dysfunction. Within Alzheimer's, you'll see as the disease progresses, a loss of memory, reasoning, language, coordination, mood, and behavior. Ultimately, a lot of people who suffer from Alzheimer's disease suffer from a loss of their own personality as the disease progresses, because they aren't able to remember things that they used to, they aren't able to remember the people around them. So it's very important that we do find ways to prevent and treat these disorders, because this is just very detrimental to somebody's mental health and way of life. So looking at the amyloid beta pathway, here we see a neuropathway, and we see the result of the dysfunction of the amyloid beta pathway. The amyloid beta pathway refers to the biological process where a protein called amyloid precursor protein, or APP, sequentially cleaved by enzymes, specifically beta secretory, to produce amyloid beta peptide. These peptides are considered a key factor in the development of Alzheimer's disease because as these peptides accumulate, they form plaques. These plaques are what we're seeing here, and the plaques cause detrimental damage to the neurological pathways. They can eventually lead to the death of these neurons. The amyloid pathway was determined to be a potential primary constituent in the Alzheimer's disease all the way back in the 1980s. So these have been under review and potential ways of treatment for a long time. So I think it is very important that we continue to do treatment on them. The target of some new treatments of the amyloid beta plaques is the mechanisms, and the technique is to focus on reducing the production, aggregation, or clearance of these amyloid beta peptides. These processes include targeting different proteins such as beta secretase or gamma secretase. Beta secretase, the inhibitors will target the BACE1, which reduces the production of the amyloid beta. If we are able to reduce the production of amyloid beta, then we will be able to reduce the amount of peptides there and thus reduce the plaques. Another one would be the gamma secretase, which modulates the design, the modulators, and they're designed to minimize toxic amyloid beta 42 production. And then a secondary option is immunotherapy, which the immunotherapy will use monoclonal antibodies that aim to clear the amyloid beta plaques. So that would be coming in and trying to get rid of some of the plaques that have already formed by targeting them to break them down. By targeting the prevention of aggregation of the molecules, the plaques can be prevented from forming. This can slow down the progression of the disorder. The next thing we'll be looking at is the tau pathway. Here we look at, we can see that this is the structure of the neuron, and then this is the non-phosphorylated tau, and this is the hyperphosphorylated tau. So, whenever tau is able to be hyperphosphorylated, it causes detrimental effects to the neuron. The tau pathway is another target of potential therapeutic approaches to treating Alzheimer's because the target of hyperphosphorylated tau and neurofibrillate causes the production of neurofibrillate tangles, and it prevents this by focusing on reducing the tau aggregation, phosphorylation, or enhancing clearance. So we can do this by utilizing kinases to inhibit the GSK3 beta and CDK5, which allows for the phosphorylation of tau. So if we are able to target those using our kinases, then we can prevent these hyperphosphorylation of tau from occurring. And then also using immunotherapy with a tau-specific antibodies to block tau aggregation or promote its clearance. So as we see, the tau is here on this neuron, and it's used on the microtubules to help keep the structure of the neuron. So we can see that this neuron is starting to dissociate, and that is because the tau is hyperphosphorylated in this condition and is able to release itself from the neuron, and so therefore the structure will not be held. And here we see the aggregation of the tau. So whenever we know that biology, neuroscience, all these very molecular things, structure and behavior go very, very hand in hand. So whenever the structure is disrupted, the function will sequentially be disrupted as well. So the normal functioning of tau. Tau is a microtubule-associated protein primarily found in neurons. It stabilizes microtubules, which are the structures essential for maintaining neural shape. So as it is able to dissociate from the microtubule, the shape of the neuron is no longer able to be held. Within Alzheimer's, we see abnormal changes in tau. The hyperphosphorylation of tau becomes excessive, and at a certain point, it causes it to become unattached from these microtubules. Then the aggregation of the tau over here allows those tau to continue going through all the microtubules of different neurons and causing the damage to just continue and continue. And eventually, you'll see a loss of function of microtubule destabilization to occur, and the tau no longer supporting the structure, and thus the cell will explode, and that will cause inflammation to surrounding cells as well, causing further damage. When tau aggregates and accumulates and spreads, it is sometimes referred to as prion-like behavior, because you will see that in a second that there's a relationship between tau aggregation as well as amyloid beta aggregation. So here we're seeing a neuron with a microtubule, and then the tau phosphorylation starting to occur and creating these helical and straight filaments. And then whenever these are produced, you have this over here, which is the tau neurofibrillate tangles. And the NFTs are very, very indicative of Alzheimer's. There is a relationship that can be observed between the amyloid beta plaques and the tau protein aggregation. While they originally independently, their relationship becomes intertwined within the disease. The alpha beta aggregation often works as a catalyst as a progression of Alzheimer's and acts as an early indicator in the presence of the disease. With its onset, the aggregation proteins reduce inflammation and they induce inflammation and also increase oxidative stress. Amyloid beta aggregates can induce tau hyperphosphorylation by activating kinases like GSK3 beta, which is one of the targets of inhibiting whenever we're trying to treat this disorder, and particularly this fragment of this disorder, and or impairing phosphatase activity. And if the phosphatases aren't able to be active, then the phosphorylation will not be regulated. The amyloid beta toxicity can promote tau misfolding and aggregation, and exposure to amyloid beta enhances the spread of the pathological tau between the neurons. And in this way, that is how this is called prion-like behavior, because as one affects the next one, affects the next one, affects the next one, that is very similar to how prion diseases are able to spread. Some other experimental approaches to Alzheimer's are protease regulators, which target protein degradation systems such as ubiquitin proteasome system, which regulates various cell processes in the immune response, antigenesis, cell proliferation, apoptosis, and DNA repair. So the ubiquitin is used to mark cells for death. So if ubiquitin isn't able to be there to mark the cell for death, then if the cell does die, the proper tools won't be recruited. As I said earlier, this will cause a necrosis type pathway to occur rather than an apoptosis pathway. And as necrosis is allowed to occur, it affects the cells surrounding in a negative way. So if we weren't able to control the ubiquitin within these types of cells, then we would be targeting cells for death, which would eventually cause the cells around it to die as well. So it would be inducing damage while trying to prevent damage. But there are lots of ways to go with different Alzheimer's techniques, and there's a lot of data that can be found within this progression of treatment. The other thing we'll be discussing is the techniques for Parkinson's disease. So, what is Parkinson's disease? We see that there are motor symptoms to Parkinson's disease, such as slowed movements, tremors, rigidity, problem with balance and walking, but there's also non-motor symptoms with difficulty swallowing, gastrointestinal difficulties, and cognitive impairment. So this is another one of those diseases that is very detrimental to a person's day-to-day life and can be very, very helpful to be treated. So the key techniques within Parkinson's disease studies include the alpha-synuclein targeting, which is basically whenever the alpha-synuclein are aggregated, the misfolding is very central to the Parkinson's disease pathway. And then using immunotherapy, we would again use a monoclonal antibody and design it to bind to the alpha-synuclein to prevent aggregation and promote clearance of the aggregated proteins. Small molecular inhibitors would block aggregation and destabilize preformed aggregates, making them be allowed to break down. And then we could use proteostasis enhancement to improve the protein degradation pathways and clear misfolded proteins. By using small molecules or gene therapy to target mTOR independent pathways, which is a master regulator of cell growth and cell metabolism, we can enhance autophagic degradation of alpha-synuclein. By using the proteasome activation to enhance the ubiquitin proteasome system, we can again use the ubiquitin to allow for these cells to be broken down in a healthy and stable way. And then we can also use chaperone-mediated therapy, where we can heat shock proteins to allow them to disfold and try to refold properly again. Or if they are unable to refold properly after being unfolded, then we can use them to target those proteins for death. And then another way for Parkinson's is using the dopaminergic pathway, because the dopaminergic pathway within Parkinson's is very, very, very vital to Parkinson's to continue and progress. So this would focus on replenishing dopamine and protecting dopaminergic neurons. Using gene therapy, we could help by delivering enzymes such as tyrosine hydroxylase to enhance dopamine production. Using TH, this is a critical enzyme to the synthesis of the catecholamines, which include dopamine. It is primarily found in the dopaminergic neurons in the central nervous system, and it is regulated with phosphorylation feedback, inhibition, and cofactors. If we were able to control the progression of these cofactors and the different feedback loops and just promote the dopamine to be produced, we could just drastically decrease the amount that these people are being affected by Parkinson's disease, as dopamine plays such a key role in the progression of dopamine, or the lack of dopamine creates such a progression of Parkinson's. So ultimately, This research is important because many individuals are struggling with these diseases, and these can cause a decrease in the person's quality of life. So if we are able to look into these proteins, look into these small molecular pieces of these diseases, and able to take them and really change them to be something to where these are not able to progress nearly as quickly, this could be revolutionary research for this field. So that is what my thesis will get more into, is different protein techniques, and really specifically how these protein techniques work at that molecular level. Thank you for listening to my presentation on different protein techniques for neurodegenerative diseases, and yes, that was all.